morning. I'm Dr. G. Vijay Pratap, lecturer in Zoology, Dr. V.S. Krishna Government Degree College. Today I'm going to teach about the general characters and classification of phylum Porifera. Initially, the name Porifera was given by one of the biologist Grant. Porifera is a Latin word which consists of two words, porus and ferro. Here porus means pore or hole, ferro means to bear. In total, Porifera means bearing of pores, which means these animals having pores all over their body. That is their special character. Initially, these animals were treated as plants, and in the year 1765, Ellis, another biologist, by observing the water currents and movements of the oscula, he mentioned that and he has given that evidence of their animal nature. Since then, they are treated as animals. Based on his observations, the great biologists like Linnaeus, Lamarck, and Cuvier placed them under Coelenter's group. After that, in the year 1816, D. Blainville created a separate group for the sponges, and he called that group Spongiaria. This group is very closer to Protozoa. At present, there are almost 500 species of sponges are available in the aquatic arena. All these animals are primitive animals and multicellular animals. Remember, primitive animals and multicellular animals. Even though they are multicellular animals, their cells do not form any organized tissues. That is why they are called as primitive animals, and that is one of their special character. Even single cell perform their duties. And all these animals are aquatic animals, and mostly these animals belong to marine region, and only one family like Spongilidae lives in freshwater. All these animals are sessile animals, sedentary animals, and plant-like animals. Coming to their body symmetry, some of them are asymmetrical and majority of them are radial symmetrical. They are in different shapes, maybe cylindrical shape, maybe branching shape, maybe vase-like, and maybe globular shape. And coming to the colors of these sponges, and they are beautiful, bright colored animals, and they may be in red, they may be in orange, they may be in purple, they may be in green, they may be in yellow, and of course some of them are dull in color like white, gray, and any other pale color. When you observe the body of these animals, their entire body is perforated by numerous pores, I told you, and canals. These pores can be called as ostia, and they are combinedly all the canals called as canal system. But one thing you remember, even though there is a canal system and ostia, but there are no organs like mouth and other physiological systems. This ostia as well as the canal system combinedly used for the purpose of their regular activities. Actually, the water enter through the astia in its body and coming out of the body through the osculum, which is a large opening of the animal. They are diploblastic animals. You know pretty well about that. Diploblastic, which means two layers. They are having two layers in its dermis. That's why the first layer is called outer ectoderm and the inner layer is called inner endoderm. In between, there is a jelly-like another layer is there that is called mesenchyme, but it cannot be treated as a third layer. The inner space of the dermis is either hollow or permeated by numerous canals. All these canals are lined with choanocytes. The interior space of the sponge's body is called spongocoel, which is one of the big cavity of their body. Actually, the skeleton-like dermis consists of either fine flexible spongian fibers, siliceous spicules, or calcareous spicules. Calcareous spicules. Almost all these animals having a capacity of intracellular digestion, and we could not find any excretory and respiratory organs in these animals because they are absent. Yes, contractile vacuoles are present but only in freshwater forms. Yes, nervous cells are there and sensory cells are there, but we cannot differentiate them. And all these sponges are monoecious or hermaphrodite. Reproduction in sponges occurs by both asexual and sexual. This asexual reproduction occurs by buds and gemmules, and the sexual reproduction occurs by ova and sperms. Yes, another important quality these sponges having is they have high potential regeneration growth. Yes, if they lost any part of its body, the body will be regenerated again within a short span of time. That is one of the highest important character of this animal. Actually, in these animals, fertilization can be done internally, and during the time of embryonic development, you could observe holoblastic cleavage, and during the time of embryo development, you could observe also the some sorts of larval forms. They may be amphiblastular larva and they may be parenchymal larva. Yes, based on the complexity of their body, sponges can be grouped into three types. The first one is ascon type, the second one is cycon type, and the third one is leuconoid type. Some of the examples for the sponges are poriferans, or cycon, spongilla, grantia, and euspongia. Now let us see the classification of Porifera. Porifera phylum can be divided into three classes. Class 1 Calcarea or Calcispongiae. Class 2 Hexacnelida or Hylospongiae. Class 3 Demospongiae. Class 1 In Class 1 Calcarea or Calcispongiae, calcare spicules are there. In class 2 and in class 3 అంటే hexactinida or hylospongia and in demospongia siliceous spicules are there. Based on that the name is given to them. Let us see the class 1 calcarea. In this class calcare spicules are present. These spicules may be monoxan or tetraxan. These spicules made up of calcium carbonate. They are in the form of calcite or aragonite. They may show asconoid or cyconoid or leuconoid complexity in its body. And they have single spongiosal cavity. That cavity opens outside through a single opening called osculum. All these are dull cut sponges. They may grow up to less than 6 inches. These sponges occur in shallow waters. These sponges having radial symmetry in its body. Choanocyte cells which are present in this class are very large. This class can be divided into two orders, order 1 Homocoela and order 2 Heterocoela. Let us see the order 1 Homocoela and order 2 Heterocoela. In order 1 Homocoela, in these animals the body wall is thin. The body wall is thin and not folded, and the spongiocele is internally lined by choanocytes. And these are all simple sponges. Example: Leucosolenia, Clathrina and Volinthus. Now let us see the order Heterocoela. Unlike Homocoela, in these animals body wall is thick and folded. Radial canals can be lined by choanocytes, and in this animal spongiocele is lined by flattened endoderm cells. You could observe leuconoid or cyconoid complexity in its body. Example: cycon, grantia and scypha. Class 2 hexactinida. These are called glass sponges. Their body have some kind of transparency, and the skeleton is made up of silice spicules. These spicules may be traction spicules with 6 rays in its structure. And in some of these animals the spicules are fused to form a lattice-like skeleton. There is no epidermal epithelium in these animals, and choanocytes are lined at finger-shaped chambers. And these all animals are cylindrical or funnel-shaped. And these animals can be found in tropical seas, and they grow up to 3 feet height. And this class can be divided into two orders, order 1 Hexastereophora and order 2 Ampidiscophora. Let us see the order 1 Hexastereophora. In this spicules are hexasters, which means these spicules are star shaped spicules. And in this order radial canals or flagellate chambers are very simple. And these animals attach to a hard object, but they are not attached by root tufts. Example: Euplectella. Coming to the Ampidiscophora. In these animals spicules are ampidisc spicules. No hexasters are present, and these animals are attached to substratum by root tufts. Root tuft is nothing but root-like structures. Example: Hyloneuma and Pheronema. Class 3 Demospongiae. It contains the largest number of sponge species. That's why it's the biggest class of the Porifera. In these animals, skeleton may be containing spongy fibers and siliceous spicules. In some animals, you couldn't find any skeleton because skeleton is absent. And these spicules are never six-rayed. They may be monoxan or tetraxan, and they may be divided into or differentiated into large megascleres and microscleres. In these animals, shape of the body is irregular and the canal system is of low complexity. It can be divided into three subclasses: subclass 1 Tetracnelida, subclass 2 Monoxonida, and subclass 3 Keratosa. Let us see the subclass 1 Tetracnelida. It can again be divided into three orders. Order 1 Myxospongida, order 2 Carnosa, order 3 Corystida. Let us see the subclass 2 Monoxonida. It can be again divided into four orders. Order 1 Hadromerina, order 2 Helicandrina, order 3 Pusillosclerina, order 4 Haplosclerina. Now let us see in detail the Demospongia class. Yes, let us start with the subclass 1 Tetracnelida. These sponges are solid and simple, rounded, cushion-like and flattened animals. They don't have any branches. In this animal, skeleton mainly contains a tetraxan siliceous spicules. Of course, in these animals, Leucan type canal system is present. In these animals, tetraxan spicules absent in Myxospongida order. Tetracnelida can be divided into again three orders: Myspongida, Carnosa, and Corystida. Let us see the order Myspongida. These are simple animals and skeleton is absent. Udaharana, example, Ascarella helisarca. Let us see the order 2 Carnosa. These are also simple structured animals, and in this group animals, spicules cannot be differentiated into megascleres and microscleres. Asters may be present in this order. Example, Placina. Order 3 Corystida. In this order, spicules are differentiated into megascleres and microscleres. Example, Geodia. Let us see the subclass 2 Monoxonida. They are variety shaped animals. Some are round, some are look like mass, some are branching type, some are elongated, some are stalked with the funnel, some are fan shaped. And in this subclass 1, in the subclass 2 Monoxonida, skeleton comprising mainly of monoxan spicules with or without spongin. These spicules may be megascleres and microscleres, and they distributed all over the world in shallow and deep water forms. In shallow and deep waters, this class can be again divided into four orders: Hadromurina, Helicandrina, Haplosclerina, and Pusillosclerina. Yes, order 1 Hadromurina. In this order, spicules may be megascleres and microscleres. Example, Cliona and Tethia. Halocandrina. These are simple structured animals. Monoxan megascleres spicules are two types: monactines and diactines are present in this animal. Microscleres are absent. Example, Helicandria and Fusulosclerina. In this order, monoxan megascleres are of two types, monactines and diactines are present, and microscleres are also present. Example, Gladeriza. Order 4, Haplosclerina. Monoxan megascleres type 1 called diactins are present. Microscleres absent. Example, Chalina spongilla. Subclass 3, Keratosa. This subclass may be known as spiny sponges. In this, body is round, skeleton composed of network of spongin fibers, siliceous spicules absent. They found in shallow and warm waters. Example, Euspongia and Hippospongia.